All right, so we're going to get started. Before I get into this whole stuff about BAF blocker and all that, I'm going to review one thing about nucleosomes, because it's very important. All right, so eukaryotic DNA is has tons and tons of base pairs. Eukaryotic DNA is very large, so in order to store it in a compact space, what we see in eukaryotes is that we have DNA wrapped around these things called histone proteins. Okay, DNA is wrapped around these things. So these are histones. And when DNA is wrapped around them, they call that unit a nucleosome. All right. Now, we know according to the central dogma that the way genes are expressed is that you have a gene in DNA that needs to be transcribed into mRNA. And then eventually that mRNA will be translated into a protein. But regarding this component here with DNA wrapped around histones, this greatly affects the ability for DNA to be transcribed into mRNA. And here's why. If a particular gene happens to be in the part of DNA that is wrapped around a histone, so let's say there's a gene right here, just happens to be right there, it will not be transcribed very well. It will not be transcribed very often because the histone is inhibiting any transcription factors from coming in to transcribe it. However, a gene that is found out here, this one not wrapped around any histones, this one's fine. This one will be transcribed very often. It will be transcribed ubiquitously. So, there are some modifications that you can do to nucleosomes to make it easier to transcribe stuff and harder to transcribe stuff. Anyone know what's the one modification you can do to nucleosomes to make it harder to transcribe something? Methylation. Methylation, exactly. So, one, if you methylate nucleosomes, if you methylate DNA wrapped around histones, there's less transcription of whatever gene that you're looking at. What's the opposite? What is the other thing? Acetylation, right. If you acetylate a nucleosome, there's more. Okay. It turns out that there's this whole series of enzymes that exist to modify chromatin, to modify nucleosomes. Some methylate, some acetylate, and some remove methyl groups and others remove acetyl groups. These are called chromatin modifying enzymes. And one of which we're going to go over in a second, I believe is called ESBAF. I don't know what it stands for, but I'm going to jump to the PowerPoint slides in a second so you see what I mean. But this one is an example of a chromatin modifying enzyme. And there are drugs that we can use to inhibit this particular enzyme, just to see the downstream effects of inhibiting it. So, to illustrate this, let's go back to the slides. That's what this thing is. Let me zoom in. All right, the top picture here is supposedly what normally happens if this enzyme, ESBAF, is allowed to do what it's supposed to do. So let's break it down. Here's our nucleosomes. We have DNA wrapped around nucleosomes here and there and everywhere. And this region in between nucleosomes is being actively transcribed. Okay? What this enzyme does normally is it keeps open regions of DNA called enhancers. I know we're not in chapter 29 yet. Technically, I think this shows up in chapter 29b. But what an enhancer region of DNA is, think of it as it's a kind of a landing pad for transcription factors. Like, how does a transcription factor know to transcribe a particular gene? Like, how does it know to transcribe something? Well, one, the gene needs to be in this nice open region, not inhibited by nucleosomes. And two, you will learn in 29b and a little bit in 29a that there needs to be a certain pattern of DNA that is right next to that gene that tells the transcription factor to transcribe the gene. It's kind of like a landing pad for the transcription factor. It knows to go to this region where there's something called an enhancer. All right, so pretend that there's just some sequence here that's really important in order to transcribe the gene up there. All right, you'll learn in 29a and b more detail about that. What this enzyme does, though, ESBAF, what this thing does is it makes sure that this region is open. And not only for this gene, but for other genes not illustrated here, that this enzyme goes around and makes sure that the enhancer region associated with various genes is open. This allows transcription factors to come in more often than they normally would because they're looking for these landing pads, they're looking for these landing sites with all these enhancer regions. And when they do, they will transcribe whatever gene that they're associated with. So that's all this thing normally does, is it keeps these things open. So what happens if you mess with that enzyme? So that's what this bottom picture illustrates here. So if you mess with this enzyme by using what's called a BAF blocker, it no longer is able to remodel chromatin. It is no longer able to mess with the nucleosomes and how they're positioned in DNA. Because of that, what was formerly this nice open enhancer region is now wrapped around, in this example here, wrapped around two nucleosomes, two histone proteins. Because it is now wrapped around, there is no longer a clear landing site for a transcription factor to come in and transcribe whatever gene. So simply because this enzyme is no longer doing its job, you lose the ability for a transcription factor to come and land here and begin transcribing a gene. In fact, what ends up happening is if a transcription factor is able to land nearby, it ends up transcribing some useless thing well, I'm oversimplifying it, but it doesn't transcribe the gene. It transcribes a thing called a non-coding RNA, which you learn about in more detail in other classes. But bottom line is it is unable to transcribe the original target gene because the landing site is now wrapped around histones because this BAF was unable to open up the landing site. All right? So you can go ahead and read all about that in details in this paragraph here, but that's all that this diagram is telling you. Yes?
For this class, how are you defining gene?
How do you define a gene, like visually or just definition-wise?
Like I would always thought it was just something, any part of the DNA that got transcribed.
Good question. There's actually some debate about that right now because of things called non-coding RNA. I believe, though, the consensus is still, as long as the gene, when it's transcribed, either leads to rRNA or tRNA, or leads to an mRNA that eventually becomes a protein, that is called a gene. There are plenty of other regions that are transcribed but don't technically make anything functional, but only relatively recently, like within the last 5 to 10 years, even those non-coding things have functions as well. And I believe somewhere in here they mentioned that. Yeah, well, one of these slides they mentioned that non-coding RNA may actually regulate stuff. But right now the consensus is still genes need to actually make something that does something. So either proteins or rRNA or tRNA, basically.
And SI and MI, because that comes from R or mRNA.
Yeah. Yeah, those wouldn't be considered genes. Those are like siRNA is short interfering RNA. It's shorter RNA than what is typically expected from a gene. Same thing with microRNA. It's shorter than what is typically expected from a gene. So it's its own weird vague category because it's still being researched right now. But consensus is still the gene is something that when it's transcribed either has a function right away, meaning rRNA or tRNA, or it'll eventually be mRNA that gets translated into a protein. That is currently the consensus on that. All right. So again, you can read the details in here if you want, but that's the main idea of what this enzyme does and what blocking this enzyme causes. Oh yeah, I guess the specific situation that they're illustrating here is that this enzyme is very important in embryonic stem cell development. Several SI sessions ago I mentioned that during organism development there's a huge shift in chromatin that occurs in order for transcription of new genes to happen during development as opposed to old genes during an earlier stage of development. They sort of talk about that again in here somewhere, but don't worry about it. All right. This is a bunch of stuff that you probably have to worry about in a genetics course, so I'm not really going to go over too much. There's dominant, recessive alleles, and linked to genes and stuff like that. That's never asked about in this class, so I'm going to skip it. Same thing here. This is stuff you might see in Bio 1 or 2, where they talk about the specific steps in meiosis, or and you know how chromosomes sort and everything. That is not asked about in this class, so I'm going to skip that. More information about inheritance, like X-linked inheritance, autosomal inheritance, dominant or recessive inheritance, important in a genetics class, not for this class, so skip. This you definitely don't need to know about, the chemical synthesis of nucleic acids, so skip, 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 skip. Okay, now finally something that you may be asked about, but it is a lot simpler than what this diagram makes it look like. So, applications, more application type questions or concepts. There's a technique in the lab called Fluorescent in Situ Hybridization, or the acronym FISH. What all this means is you can design a fluorescent probe that will specifically bind a particular DNA or RNA sequence that you want to investigate. Let's say there's a mutated gene that you know that if someone inherits this mutated gene, they're going to have some debilitating genetic disease in a couple years. So you need to be able to see if this person's going to have that disease so you can prepare treatments and things like that. What they often do now is they pre-design these probes that specifically will only bind to the mutated version of a gene. So what ends up happening is these probes, and that's not necessarily what this picture is illustrating, but for the example I'm talking about, this probe, you apply it to some DNA sample, and if it shines brightly under a microscope, if it shows a particular color under a microscope, it means it has found the mutated copy of the gene, and that person, that DNA sample is from a person who is very likely to develop that particular genetic disease. All right, so that's one huge application of this thing. The bottom line of how it works is that there's a probe that binds to a target DNA sequence and releases some color. Okay. All right. Other interesting things, talking about chromosomes and such, is this very weird chromosome abnormality called a Philadelphia chromosome. There's a lot of detail on here, but basically how this works is, you know how we have, 23, 26, I don't even remember anymore. We have a certain number of chromosomes that you have to remember in the genetics course that I don't remember. Sometimes two different chromosomes can spontaneously fuse together. And in this case, the fusion of two chromosomes here, of two parts of two chromosomes here, actually creates a gene that normally doesn't exist. Okay? So if you read the details here, you'll see that, if you look at the ABL gene in one chromosome and a BCR gene in another chromosome, what the Philadelphia chromosome is an accidental combination of these two completely separate genes. When they combine together, they end up forming a gene that when it is transcribed and it is translated, creates this thing. This is the main feature of the Philadelphia chromosome. It creates an uncontrolled tyrosine kinase. So tyrosine kinase, and you see the phrase kinase, you know that this enzyme is going to go do what? phosphorylate something, right? That's bad, especially because it's uncontrolled. That's the bad part. This enzyme's going to go along and phosphorylate everything it possibly can. That is usually associated with uncontrolled cell division. When cells divide, there's this process of activating certain enzymes that are required for cell division. These activation steps are usually associated with controlled kinases, so that the cell only replicates when it's ready to. However, due to the Philadelphia chromosome, this accidental fusion of genes, creating a gene that normally doesn't exist, you create a kinase that goes and phosphorylates everything everywhere all the time. So the cells that this Philadelphia chromosome is in are going to end up replicating over and over and over again non-stop. We know that that condition is associated with cancer, and that is indeed what the Philadelphia chromosome leads to. It's some type of blood cancer. Is it on here? I can't remember what it's called. I don't remember, and it's not on here, so I guess you don't need to know, but it is some type of blood cell cancer. Anyways, there are some drugs designed to combat this issue by specifically targeting this unusual enzyme that is only made in a Philadelphia chromosome. You might see this term used in various drug commercials and things like that. This is the scientific name for it, or the company name for it. All it does is it inhibits this uncontrolled tyrosine kinase. So that's all the information that's given about the Philadelphia chromosome. Okay. Oh yeah, competitive inhibition. Anyone remember what that means? What does it mean? Where does this bind to? Competitive.
Covalently binds to any site but the other.
Competitive means it binds to the active site. Covalently, you might be thinking of like suicide inhibitor or something like that. Yeah. But that's fine. He doesn't usually ask about that anymore, but it's just a little bit of a review. Competitive means whatever this thing will do, it will bind to the active site of a particular enzyme. This is, a mix of the previous two slides. You can design FISH probes, fluorescent in situ hybridization probes, to specifically look for the Philadelphia chromosome. Some more interesting pictures of applying fluorescent in situ hybridization. Some interesting, one last interesting fact they talk about is what sometimes happens when cancer cells are allowed to replicate over and over again. Normal cell replication usually is associated with an even split of chromosomes between two daughter cells. However, when cancer cells are replicating over and over and really quickly, sometimes the chromosomes start sorting in different amounts. Like for example, instead of evenly splitting 23, is that right? Did I get that right? 23, all right. Instead of evenly splitting 23 chromosomes between two daughter cells, you might get 20 in one and 26 in the other one. They usually call that, polyploidy or something like that, or maybe aneuploidy is the good one. But I can't remember. It's not on here. It's a genetics term anyway. But what this slide is trying to tell you about is mostly that when cancer cells replicate, you lose the stability that chromosomes have. They use terms like aneuploidy, genomic stability is normally what you see, and when cancer cells replicate over and over again, those are some of the first things that are lost. Anyone recognize the term P53 in other classes, right? So if you don't know what P53 is, that's kind of one of the main components of this slide here. P53 is known as a tumor suppressor gene, meaning, and it's in the name, what this gene does is it helps prevent the development of cancer. It does so by regulating these components it's like a checkpoint thing. It regulates all those checkpoints that you might have learned in an earlier biology class. Like there's a checkpoint between G1 and S, there's a checkpoint between S and G2, a checkpoint between G2 and mitosis, and so on and so on. Turns out this one gene actually regulates all of that and more. P53 is like the most important tumor suppressor gene that you have. And therefore, a lot of cancers, one of the first things you end up finding in it when you analyze the genome of a particular cancer is that something is wrong with the P53 gene, okay? It doesn't mean that all cancers are caused by a mutation in p53, because sometimes p53 is mutated only after cancer occurs. But when you analyze a cancer genome, you will find a mutation in p53. There's debate over whether p53 is messed up first before cancer happens, or cancer causes the mess up of p53. Again, that's probably way more detail than you need, but that's the context given with these slides here. These are applications and neat, interesting facts about chromatin and chromosomes. And that's the end of chapter 11. So, 11's really short, as you can see. And I assume Jesus is in 28 now, right?
You just finished 11.
Oh, he just finished on. Did he at least start 28?
Yeah.
All right, well, good. We'll get a little bit ahead then. All right, because now you've learned about the components of DNA, the nitrogenous bases, the nucleotides, and you learned a little bit of how when they're put together, you form DNA double helices, you form chromatin, you form nucleosomes in eukaryotes. In prokaryotic DNA, it's circular instead of linear. You're starting to put things together. Now we get into the actual mechanisms behind that. And again, you learn about transcription in chapter 29, but you also heard a little bit about what transcription is and how that's important. But first, DNA replication. So, let me see where they choose to start this. Blah, 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 history stuff, a picture of DNA replication. So, actually, let me see if there's another picture that's better. Yes, this one's better. All right. Way back, long time ago in the ancient history of biology, they didn't know how DNA was replicated. There was 3 theories with how DNA was replicated. Via something called conservative replication, something called semi-conservative replication, and something called dispersive replication. They had no idea. So they had to figure out an experiment to figure out how DNA was replicated. We're kind of spoiled a bit. We might know the answer here. Does anyone know of these 3 how DNA actually is replicated? Semi-conservative, right? It actually is replicated semi-conservative. So let's look at that one first, since we may have learned that in a previous class. The theory of semi-conservative replication, and it has been confirmed, is that you have 2 strands of DNA. If you want to replicate it, how is it done? Step 1, you separate the 2 strands of DNA so that they are 2 individual strands. Follow this lighter color, this I guess white colored strand. They are 2 separate strands here. Replication occurs by using each individual strand of DNA as a template for a new strand of DNA. All right, so this new DNA is this orange stuff here. So basically we have a bunch of enzymes that will read each individual template strand and understand and know what base is supposed to be added there to form a complementary DNA sequence. We've kind of already practiced this at SI, right? That if we're given one DNA strand, you are asked, well, what's the complementary DNA strand? Well, enzymes do that all the time in order to replicate DNA. So again, the theory of semi-conservative replication is you separate double-stranded DNA into 2 template strands that each can be used as a template to replicate a new strand of DNA. And that process is repeated over and over again. Okay? Well, what's the theory with conservative replication? So again, back when no one knew how this worked, basically the theory with conservative replication is there was no separation of DNA strands from each other. I kind of think of what conservative replication is as if you will photocopy the DNA. So notice that you don't separate it into pieces or anything. You literally just take it and copy it as a whole like that. That was one theory of how DNA was replicated, is by photocopying it via conservative replication. There is no separation of strands of DNA, so notice that the very original strand of DNA is still there in the 2nd phase here and still there in the 3rd phase here as well. So that's the conservative theory. Dispersive theory is pretty crazy. Dispersive theory is a little bit more disordered than the semi-conservative theory. Semi-conservative theory is you just nicely unwind the 2 strands of DNA from each other. Dispersive theory is you break it into pieces, into a random assortment of pieces, the fragments of template DNA all over the place. And then you kind of just hope to piece them together with new DNA, and that's sort of what they're illustrating here, is that the new DNA represented in orange is pieced together in these random assortments here, and you create your two new versions of DNA here, and you do this over and over again. So dispersive is basically you shatter your template DNA into pieces, and you mix them back up together with new DNA. That was dispersive DNA replication. So visually we can see what it would look like, but in terms of the lab, we can't see DNA doing this. So how did they figure out which one was which? They performed an experiment where they used a radioactive isotope. I believe the experiment is called the Meselson stall experiment, which you are sometimes asked about on the exam. The Meselson stall experiment. The way it works is you have old DNA and new DNA defined by particular isotopes. Basically, you see this lighter colored DNA is defined as old DNA in the experiment. It is made with nitrogen 14. Okay, it is made with nitrogen 14. Well, where does the nitrogen come from? Nitrogenous bases. Your DNA is made of nitrogenous bases. So what they did is they had DNA with nitrogen 14. When they forced the DNA to replicate, either by using enzymes or whatever, when they forced DNA to replicate, they made sure that the only nucleotides that could be used to replicate new DNA would be nucleotides labeled with nitrogen 15. So everything you see in orange on this diagram is labeled with the radioisotope nitrogen 15 instead of nitrogen 14. With these two different radioisotopes, you can keep track of how DNA is used to be replicated. All right, so you'll see in a second. I believe it's actually I will have to draw this, so I'm going to have to flip back and forth from the slides. So let's see what it would look like if we did conservative replication. So with conservative replication, with zero amounts of replication, all we have is DNA composed of nitrogen 14. Okay. After one round of replication, based off our illustration here, we have DNA that is composed of only nitrogen 14 and DNA that is only composed of nitrogen 15. This subtle difference you can actually see in the lab when you centrifuge these DNA samples down. What it'll come out as, is that for conservative replication, if we had 0 rounds of replication, 0 rounds, we said earlier that all you get is DNA that is labeled with nitrogen 14. So when you centrifuge it down and you have this test tube here, what you're going to find is a single line representing DNA made with nitrogen 14. Okay, that's with 0 rounds of replication. With conservative replication, if you actually did that one round of replication, I'll flip back to the picture so you see, so you're reminded again what happened. You created a DNA sample that is only made of nitrogen 14 and a DNA sample that is only made of nitrogen 15. What that will look like in a test tube, so after one round, what that would look like in a test tube, this is another way of describing this is a densitometry analysis, because you're separating things by density. You should see 2 distinct lines. One representing all of the DNA that is made with nitrogen 14, and a new line that came out of nowhere that is a little bit lower in the test tube because it's heavier, DNA representing ones made with nitrogen 15. Okay? So this is what conservative replication, this is what your test tube will look like after one round of replication. Just for the heck of it, what would 2 rounds of replication look like? Well, let's go look at the picture. And again, we're kind of cheating because they had no idea which of this was going on. All they had was the test tube part. But after 2 rounds of replication, we find a DNA sample just with nitrogen 14 and 3 copies of DNA made with nitrogen 15. So what would that look like over here? We'll still have that one band up here representing old nitrogen 14. And this time, the band representing nitrogen 15 might be a little bit darker. Why? Well, because we have 3 times the amount of new DNA that was made, 3 times the amount of DNA that is made with nitrogen 15, 3 times the amount of DNA that's orange in that picture there. So, if they did this experiment and they found that in their test tube after 1 round of replication there was 2 bands here, and after 2 rounds of replication there was 2 bands with one of them getting darker, they would have been able to conclude that DNA replication proceeded through a conservative pathway. All right? For illustrative purposes, let's figure out what the test tube should have looked like, because we already know the answer is that DNA proceeds through semi-conservative. Okay, again, sometimes this is asked about in a word problem format, this Meselson stall experiment, but we'll finish covering this and I'll give you some potential questions. So semi-conservative, let's see. 0 rounds of replication, it looks like it's the same. It still is only going to be made of nitrogen 14. After 1 round of replication, we have something different. After 1 round of replication, we've created 2 hybrids, 2 hybrid strands of DNA. Each of these is composed of 1 strand of nitrogen 15 DNA and 1 strand of nitrogen 14 DNA. So this is hybrid. What that will look like, well, is after zero rounds of replication, we still have that 1 band representing nitrogen 14. After 1 round of replication, we will have only 1 band show up. This band is going to be directly halfway between the band that we expect for nitrogen 14 and the band we would expect for nitrogen 15. So this 1 band that shows up is representing that hybrid DNA that we saw in the picture, where each sample of DNA there is half of nitrogen 14 and half of nitrogen 15. So you end up with just 1 densitometry band in your test tube. Okay? To continue on, let's see what happens after 2 rounds of replication. After 2 rounds of replication, we have a slight, a different thing now here, is that we still have 2 copies of DNA that are the hybrid type, but now we have 2 copies of DNA that are only made of nitrogen 14, only made of nitrogen 15. Why is that? Well, again, the theory of semi-conservative replication is you can only use each individual strand of DNA as a template for the next one. So up here, the orange band that was made in this step right here now has to act as a template in this 2nd generation of DNA. That's why you get 2 copies of DNA that are only made of nitrogen 15, is because the template was 1 strand that was nitrogen 15. All right? So what will that look like? Well, if we have 2 different versions of DNA, that should show up as 2 lines in our test tube. One now represents a type of DNA that's only made of nitrogen 15, and the other one will be halfway in between where we would expect nitrogen 14 to be and where we expect nitrogen 15 to be. So this is still the hybrid line right here. This band will show up representing the hybrid line. Okay? So again, for semi-conservative replication, we started with just DNA made with nitrogen 14. Then when we did 1 round of replication, we've created hybrid DNA, so we have this band that's kind of in the middle here. And after 2 rounds of replication, though, we have that band that's still the hybrid DNA, but now we have a band representing only DNA made with nitrogen 15. Okay, so this is indicative of semi-conservative replication. What would dispersive look like? Dispersive will start the same way as everything else did. 1 band of nitrogen 14. And sometimes this one's a bit hard to explain, but I'll try my best. Dispersive replication, remember, is when you shatter DNA into pieces and you put them back together. When you put them back together, you create hybrid DNA again. And in fact, this ratio, although it's kind of hard to see, is still 50% nitrogen 14 and 50% nitrogen 15. So you do still create hybrid DNA with dispersive replication. What that will look like is 1 band in the middle here between where we would expect nitrogen 14 to be and where we would expect nitrogen 15 to be. We create a band right in the middle. So notice that after 1 round of replication, we cannot tell the difference between semi-conservative and dispersive replication. So we have to do it again. We have to do a 2nd round of replication. what ends up happening, I'll draw it first and then we'll go back to the picture here, is that after a second round of replication, you will get only one band. It will just be a little bit closer to the N15 side. Okay, you will always only, with dispersive replication, you will always only get one band in your test tube. Because, when I go back to the picture, dispersive replication will evenly distribute the fragments of DNA between all made samples. So it will evenly distribute all the N15 fragments of DNA, it will evenly distribute all the N14 fragments of DNA, so that all 4 copies of DNA will have the exact same amounts of N14 and N15. Because of that, you only get one band that shows up. You only get one band that shows up in your test tube. So these are your 3 possible arrangements here. On the exam, you may be asked, what would Meselson and Stahl have observed if it turns out DNA was replicated via conservative replication? Well, they would have observed 2 bands after 1 round of replication, and still 2 bands after 2 rounds of replication. And they may even go into more detail in the answer choice where they say, well, one band will be a lot darker than the other one. So it'll be a descriptive type question if this was asked on the exam. They might ask, what would Meselson and Stahl have observed if DNA replication was dispersive? Well, that's pretty easy. No matter how many rounds of replication you do with dispersive, you will always only get one band in your test tube because they evenly distribute the fragments of DNA. So they knew, though, in real life that DNA was semi-conservative, and they figured it out when they did this experiment, and they got these exact patterns where after 1 round of replication, they have 1 band, but after 2 rounds of replication, all of a sudden 2 bands show up. Okay? That's how they figured out it was semi-conservative. So that's what you need to know for the Meselson-Stahl experiment, what it proved, and potentially what alternate history we could have had if they discovered something else. All right. Let's see if there's anything else. Yeah, those drawings that I had there with the test tubes and everything, this is just a mathematical, graphical version of that. You won't be asked about that, so I'm just going to move down from that. And now we get to some features of DNA. All right.
Can you go back to the picture before, please?
This one?
No, the other one.
This one.
Are they showing that they had N15 first and then they added N14?
Yeah, maybe. I might have said it backwards this whole time. My bad. Who cares?
No, I don't know if I learned it backwards or we said it backwards.
Maybe I said it backwards. It seems like if this is the way it went, I said it completely backwards. My bad. It probably does make sense that they would probably use only radioactive DNA for the original strand. All right, well, who cares? Change everything. Erase everything. Everywhere you see a 14, put a 15. Everywhere you see a 15, put a 14. Yeah.
I was making sure because I thought I learned it. I'm not sure which way it's supposed to go now.
It is this way. I just, for some reason, thought of it the other way. But you're right. It doesn't matter. The bottom line is there's 2 different types of DNA.
It's just which band will appear, which I was getting a little...
Yeah, I probably mixed them up. That's fine.
Sorry, I'm not trying to call you
Flip
out.
everything around. No, it's fine. Flip all your test tubes upside down. Everything's correct. It's just the wrong order. All right? Everything is correct. It's just the wrong order. It's just everywhere there's N14, it's N15.
Can we say that in the text?
Yeah, sure, why not? No, it won't be that specific. They won't ask like how many N14 bands, N15 bands. It won't be like that. It'll be literally how many bands do you see, 1, 2, or 0. All right. All right, features of DNA. Make sure you know these features. This applies, both of these features here, the phrase bidirectional and semi-discontinuous, apply not only to prokaryotes, but it applies to eukaryotes as well. Prokaryotes is just an extra little thing to worry about. So what does each mean? All right, let me go to a nice picture to illustrate what each one means. So, the phrase bidirectional, DNA replication is bidirectional. The way what this means is, in order to replicate DNA, you have to open up your template DNA. So here's an example here. You had double-stranded DNA, and what they've done is they opened a hole in double-stranded DNA. This is called the origin of replication. You open up an ORI-C site is sometimes also used, an origin where enzymes will begin replicating DNA. It is bidirectional because when you open up a single hole in DNA, you can go to the right in this picture and you can go to the left in this picture to replicate DNA. That is what they mean by bidirectional. I'm pointing to the wrong thing. But that's what they mean by bidirectional, is that you can go to the right and you can go to the left. DNA replication is most commonly bidirectional. I know there's some prokaryote that does only unidirectional replication, but we don't care about that in this class. That's just a microbiology genetics thing. In this class, we associate the phrase bidirectional with DNA replication. Okay, the other phrase, and I believe there's another slide to describe this, the other phrase associated with DNA replication is the phrase semi-discontinuous replication. This phrase can be applied to each origin of replication. What that means is, so here's opened up DNA, and in this picture, not shown, there's an enzyme at the front end here. Actually, it's shown down here, my bad. There's an enzyme at the front end of DNA, a series of enzymes that are replicating the DNA strands and simultaneously opening up the template DNA to replicate it over, to continuously replicate it. Now, the reason why we use the phrase semi-discontinuous is that, you'll see, probably it'll have to be next SI session, you'll see that one strand of DNA can be made continuously. That strand of DNA is called the leading strand of DNA. This has to do with way back, I think, in chapter 10a, I told you about 5' to 3' stuff, right? To prepare you for next SI session, go back to that and see what it said. Because of the rules that I gave you back then, one strand of DNA can be made continuously. However, the other strand of DNA, because of the limitations in direction, can only be made discontinuously. Therefore, when you put those two strands together, we use the phrase semi-discontinuous, where it's partly continuous, where one strand of DNA is made continuously, and the other one has to be made in little fragments called, and you might remember from Bio 1, Okazaki fragments. DNA is made discontinuously. All right. I'll probably have to talk about that next time because I know someone's using this class in a second. Before that, let me see if there's any other extra little facts I can give you. All right, to help you prepare for chapter 28, again, go look at those directional rules that I gave you a long time ago. You might find that enzymes can only replicate DNA in a can only make new DNA in a 5' to 3' direction, and they can only read template strands of DNA in a 3' to 5' direction. I'll state that again when I go over this in detail next SI session, but you go look in your notes, I've said it before. Other stuff to help you prepare, you will need to know the facts associated with the enzymes DNA polymerase 1 and, where is it, DNA polymerase 3. DNA polymerase 3, so make sure you know the different components that are composed of each one and the functions that each one has. That will be covered in detail. That is guaranteed to be asked about on the exam. All right, that's it for now. If anyone has any questions, I'll be up here for a little bit before the next class comes in.